Hey, hello everyone. Hope you all are well in this lockdown period. This is Preeti. I'm pursuing research in the field of chronobiology. Let's try learn something new today. Yes, something which is related to us only. Fine? So for this, today we will talk about the cell. I know. Now you all are thinking that what is new in this cell, right? For this, we will see the new prospects to learn things related to cell that you know already. Okay? In this slide, you see that what is cell? I mean the definition of cell. So actually cell is the fundamental unit of life. And amazingly, all life begins as a single cell. Yes, only a single cell. So, if you people are interested in life, you must know about each and every single thing related to cell. As Lamarck stated that nobody can have life if its constituent parts are not formed of cells. Okay? Let's begin with the background literature of the cell. Robert Hooke studied a piece of cork which have a honeycomb-like structures. He named these compartments as a single or cellule structure, now known as cells. After Robert Hooke, Leeuwenhoek was first to observe, describe and sketch a free life cell. Okay, here you see two emojis which shows smiling face. Now the question is why they are smiling? Because smiling means something living in nature, right? Likewise, living related to cells, right? So, I hope you all are able to correlate the things like why it is important to study a eukaryotic cell. Shall I move to the next slide? Okay. These are few points about a cell. Actually, cell contains an organized nucleus and a membrane covered cell organelles. In this, protoplasm is called protoplast, which composed of cytoplasm, nucleus and vacuoles. In this, cytoplasm has a complex organization formed of cytoplasmic matrix and cell organelles. There are cytoskeletal structures which not only provide movement to the cytoplasm but also other locomotor activities which are necessary for the successful survival of the cell. Genetic material is organized into chromosomes and chromatin. This is the most significant structure which are present in eukaryotic cells. Usually centrioles are present in eukaryotic cells. So here you see the most important and significant part of the lecture, that is the ultrastructure of an eukaryotic animal cell. In this slide, we will talk about all the structures and their functions of cell one by one. Okay? Let's start with microvilli. These are microvilli. Okay? Actually, microvilli are finger-like evaginations which are engaged in absorption. They increases the surface area of the membrane, right? Usually they are present in cells like intestinal and uriniferous tubule cells. Fine? Next is tight junction. When the membrane of two adjacent cells are closely or tightly attached to each other, they form tight junctions. Next is terminal bars. These are bands of thickening occurs on the inner side of the membrane. As I already told you that the centrioles are present in eukaryotic cell. The next one is interdigitations. They increases the area of contact between two cells for the exchange of materials between the cells. Okay? Here the nucleus, chromatin, nucleolus, nuclear envelope, these all are the parts of a nucleus. Okay? Let's move to the desmosome. When the adjacent membrane possesses disc-shaped thickenings, they form desmosomes. They function as spot wells. Next is gap junction. Actually, gap junctions are when the adjacent cells connected to special proteins named as connexons. These are especially special proteins which we called as connexons. They form gap junctions. This is the cell membrane of the adjacent cells, right? Here you see exocytosis. Actually, exocytosis is a process. This is the process of when some materials or cell debris which are no more important, no more helpful for the survivor of the cell, they move outside. Right? So the process is called exocytosis. This is plasma membrane. This plasma membrane actually a covering which covers the cell to the outside. Protective in nature. Okay. This is endocytosis. This is just opposite process of the exocytosis. As you see, this is endo, means the movement of materials from outside to inside. And those materials which are necessary for the successful survival or which may help in metabolic process of the cell goes inside cells in the form of endocytic vesicles. Next is Golgi apparatus. I know you all are very much aware of this Golgi apparatus or Golgi complex. Actually, these are the post office of the cell, right? Because Golgi apparatus helps in translocation of the material to their final destination, right? This is lysosome. Lysosomes actually also known as suicidal bags, residual bags. Okay? This is mitochondria. Most interesting thing in the eukaryotic cell because they are the powerhouse of the cell. Why it is powerhouse? Because it generates power in the form of ATP, that is adenosine triphosphate, which again is a energy currency of the cell. Got it? Next is free ribosomes. Here you see SER, that is smooth endoplasmic reticulum. These rod-like structures, microtubules. Here you see peroxisomes. Okay, the question arises that what is the function of peroxisomes? Actually, peroxisomes contains oxidative enzymes. You may ask a question that what is the function of oxidative enzymes? So the answer is xenobiotics or foreign particles which cannot be metabolized by normal enzymes broken down inside peroxisomes. So this is the important function of the peroxisomes. Here you see microfilament. This is RER, that is rough endoplasmic reticulum, which modifies protein or helps in protein synthesis. Again, this is also a most important and significant part of this cell or cell organelles. So this is all about the ultrastructure of an eukaryotic cell. So this is all about the eukaryotic animal cell. Hope this will help you out for your better understanding things related to cell. Thank you so much for your patient learning and listening, of course. And your questions, queries are always welcome. You can give your suggestions if any. So, thank you so much. Thank you.